Hello my dear students, welcome back to Biology at Priyajit. In today's class, we will study one of the most important topic in microbiology and environmental biology, that is microbial nitrogen cycle. Nitrogen is the essential for life because it is a major component of amino acids, proteins, nucleic acids, chlorophyll and many other cellular molecules. Although our atmosphere contains about 78% nitrogen gas, most organisms cannot use atmospheric nitrogen because of its extremely stable N triple bond N composition. So microorganisms solve this problem by continuously converting nitrogen between different chemical forms. The four major processes we will focus today on are ammonification, nitrification, denitrification and biological nitrogen fixation. So let's begin. Ammonification. Let us begin with ammonification. Ammonification is the microbial conversion of organic nitrogen into ammonia or ammonium ion during decomposition. It is also referred as mineralization of organic nitrogen. Microbial proteases first break proteins into peptides and amino acids. Amino acids then undergo deamination, releasing ammonia. So the basic sequence is protein will be converted into peptides, then into amino acids, then it will convert into ammonia, then it will convert into ammonium ion. So at physiological pH, much of the ammonia exists as ammonium, that is NH4 plus. There are many heterotrophic bacteria, fungi and archaea participate in this process. So the genera such as Bacillus, Pseudomonas, Proteus and Clostridium are the examples. Ammonification is therefore the important link between organic nitrogen and inorganic nitrogen. So this is the image showing the process of ammonification. Now we'll go to the nitrification. Nitrification is the next process. It is the biological oxidation of reduced nitrogen, mainly ammonia or ammonium, into the nitrite and then nitrate. Traditionally it occurs in two steps. Step 1 is ammonia oxidation. NH3 will be converted into NO2 minus. The enzyme ammonia monoxygenase or AMO initiates ammonia oxidation, while hydroxylamine is subsequently oxidized into nitrite. Important ammonia oxidizers include nitrosomonas and nitrosospira. Interestingly, ammonia oxidation is not restricted to bacteria. Ammonia oxidizing archaea, particularly members of Nitrosospherota, are important in soils, oceans and nutrient poor environments. So there is also a fascinating modern concept called uh, COMAMOX or complete ammonia oxidation. Certain nitrospira can perform the entire process, that is NH3 to NO2 minus to NO3 minus within a single organism. Step 2, nitrite oxidation, that is NO2 minus to NO3 minus. This step is catalyzed by nitrate oxidoreductase or NXR. Important nitrite oxidizers include Nitrobacter, Nitrosospira, Nitrococcus and Nitrospina. Nitrification is important for nitrate production, soil fertility and wastewater treatment. However, intensive nitrification can also contribute to soil acidification. Denitrification. Now we come to denitrification. Denitrification is essentially the reverse direction of nitrogen oxidation. It is a microbial respiratory process in which oxidized nitrogen compounds are progressively reduced, ultimately producing nitrogen gas or nitrogen N2. So the classical sequence is NO3 minus to NO2 minus to NO to N2O, then nitrogen. It generally becomes important under low oxygen or anoxic condition. The major enzymes are nitrate reductase, nitrite reductase, then nitric oxide reductase, and ultimately nitrous oxide reductase. So they are commonly represented as NAR, NAR, NAP, NAR, NAP, that is NARS, NARK, and then NOR, then NOSZ. So the final enzyme is NOSZ, which converts nitrous oxide into nitrogen gas. This step is particularly important because N2O by nitrous oxide is a potent greenhouse gas. Thus, complete denitrification returns nitrogen to the atmosphere as relatively harmless nitrogen, whereas incomplete denitrification may release NO and N2O. So this is the image showing the process of denitrification. Biological nitrogen fixation. Now let us discuss perhaps the most fascinating microbial process, biological nitrogen fixation, or also known as BNF. BNF is the enzymatic conversion of atmospheric nitrogen into ammonia NH3 by microorganisms containing the enzyme nitrogenase. So the overall reaction is N2 8H plus 8 electron plus 16 ATP. It will form 2NH3 plus hydrogen plus 16 ADP plus 16 Pi. So why does this reaction require so much energy? Because N triple bond N is extremely stable, so the enzyme responsible for nitrogenase, the conversional molybdenum nitrogenase contains two major components. First is the iron protein encoded by NIF-H. It contains 4 Fe4S clusters and transfer electrons to the second component. Second, the MOFE protein encoded by NIF-T and NIF-K. It contains the P cluster and the catalytic FeMO cofactor where nitrogen reduction takes place. The simplified electron flow, electron donor will form the Fe protein, then MOFE protein, and ultimately nitrogen. So each electron transfer cycle requires ATP hydrolysis, making nitrogen fixation one of the most energetically expensive microbial processes. So here the image showing the basic process of biological nitrogen fixation. Alternative nitrogenase. An important point for undergraduate or postgraduate student is that nitrogenase that is not restricted to molybdenum. So there are three major systems: MO nitrogenase or NIF-HTK, V nitrogenase or VNF-HTGK, and Fe only nitrogenase or ANF-HTGK. When molybdenum is available, ammo-nitrogenase is generally preferred. Under molybdenum limitation, microorganisms may use alternative V or Fe only nitrogenase depending on metal availability. This is an excellent example of the metabolic flexibility of microorganisms. Why must nitrogenase be protected from oxygen? Here is another crucial concept. Nitrogenase is highly sensitive to oxygen. Therefore, nitrogen fixing microorganisms have evolved different protective strategies. For example, Clostridium fixes nitrogen anaerobically. Azotobacter maintains a very high respiratory rate to consume oxygen rapidly. Cyanobacteria such as Anamina and Nostoc use specialized cells called heterocysts. And in legume root nodules, leg hemoglobin helps maintain a low concentration of free oxygen while allowing respiration to continue. NIF genes and their regulation. The term NIF refers to genes associated with nitrogen fixation. The three core structural genes are NIF-H, that is Fe protein, NIF-T, MOFE protein subunit, NIF-K, MOFE protein subunit. So together NIF-HDK encode the core ammo-nitrogenase, but nitrogen fixation requires many additional genes. For example, NIF-E and NIF-N participate in FeMO cofactor assembly. NIF-B functions in early FeMO cofactor biosynthesis. NIF-U and NIF-S are associated with iron sulfur cluster assembly. NIF-V is involved in hemocitrate synthase, and NIF-A acts as an important transcriptional activator of nitrogen fixation genes. In many diazotrophs, NIF-A works with delta 54 dependent RNA polymerase to activate the transcription process. So nitrogen fixation is generally suppressed when fixed nitrogen, particularly ammonia, is very much abundant. So under nitrogen limitation, appropriate regulatory systems activate nitrogen fixation pathways. In Klebsiella pneumoniae, for example, the NIF-L NIF-A system plays an important regulatory role. Legume rhizobium symbiosis. One of the best known examples of biological nitrogen fixation is the relationship between leguminous plants and rhizobial bacteria. The process involves bacterial recognition, root hair attachment, node factor signaling, root hair curling, infection thread formation, and finally nodule development. Inside the root nodule, nitrogenase converts nitrogen into ammonia, so the plant supplies the bacteria with carbon compounds and energy, while the bacteria provide reduced nitrogen to the plant. Leg hemoglobin helps maintain the oxygen balance required for both respiration and nitrogen fixation. The ammonia produced is rapidly assimilated mainly through GS-GO-GAT pathway, producing glutamine and glutamate that serves as major nitrogen donors for biosynthesis. Summary: let us integrate the entire microbial nitrogen cycle. Organic nitrogen after ammonification will form the ammonium, then ammonia oxidation it will form the nitrite oxidation, nitrite, then it will form the nitrate, then denitrification, then NO to N2O, then nitrogen. So microorganisms act as the biological engines of the nitrogen cycle. So at the same time we can say that atmospheric nitrogen, with the help of nitrogenase enzyme, it will convert it into ammonia or ammonium, then will assimilate into organic nitrogen. So let us remember the four processes in one simple sequence. Ammonification releases nitrogen as ammonia, nitrification oxidizes ammonium to nitrite and nitrate, denitrification reduces nitrate and ultimately returns nitrogen to the atmosphere as nitrogen, and biological nitrogen fixation brings atmospheric nitrogen back into the biosphere as ammonia. So my dear students, understanding this microbial process is essential not only for microbiology but also for soil fertility, agriculture, wastewater treatment, ecosystem functioning and global environmental change. Thank you for watching. If you found this class helpful, useful, please like the video, share it with your friends and subscribe for more UG PG level biology lessons.